Good afternoon, students. Today we are discussing pure line selection. Okay, in the last lecture we have started to discuss pure line selection. Okay, in the last lecture we have discussed the concept of pure line and also the fill technique which is used for the pure line selection. Okay, and in today's lecture we are going to discuss the precautions which we have to take while practicing the pure line selection on the field. And another important thing we today will discuss regarding the advantages and also the limitations of the pure line selection. Okay, today's our lecture it mainly deals with the precautions which the plant breeder have to take while practicing the pure line selection in the field, and also the advantages and the limitations of the pure line selection. Okay, though we have discussed the pure line selection, the concept and the field technique in the last lecture, here the sum of the summary we can say. Yes, pure line selection. Here, the term pure line, it was firstly coined by the W.L. Johnson, a Danish botanist who was working on the princess variety of the French bean in the 1903. Okay, and the pure lines, these are nothing but the group of plants which are all obtained from a single self-fertilized homozygous plant. Okay, pure line, these are nothing but the group of plants which are obtained from the single self fertilized homozygous plant. Then also we have discussed the characters of the pure line. Pure lines, these are nothing but the progeny of the single self fertilized plants. And another important thing, as these are the progeny of the single self fertilized homozygous form, these are all genetically uniform. Their genetical constitution of all the pure lines is similar. Okay, as the genetic constitution is similar, that means all the pure lines are identical in their phenotypic characters. Okay, but there are certain minor differences which are arised due to the environmental variations. Okay, these are the characteristics of pure line we have discussed. Then also we have discussed the production of pure lines. As the self pollinated crops, these are naturally homozygous, and that's why the it is natural to it is very natural to find the pure lines in the self pollinated crops. But in cross pollinated crop, as these are naturally heterozygous, these are genetically variable, that's why these are subjected for the self pollination for the number of generations. Okay, and that's why the pure lines which are produced in the cross pollinated crops are said to be the inbreds. Then the pure line selection, that is the definition of the pure line selection, that is in this case the large number of the plants are selected from the population of self pollinated crop. Okay, then these crops are harvested individually and their individual progenies are evaluated, while the best progenies, these are released as the new pure line variety. Okay, then about the fill technique we have discussed the pure line selection. It is the method, it is generally practiced for the crop improvement of self pollinated crops. Okay, it is generally applied for the self pollinated crop. Okay, in the summary you can say this is the mixed population of the self pollinated crop, that is the farmer's field or the research station from which the plants are selected. Okay, where the selection is carried out. Then these plants, these are harvested individually, their seeds are collected, and the seeds of each of the plant, these are kept separately. Okay, that is completely opposite to the mass selection. Here the seeds are not mixed with each other. Okay, the seeds of the each of the plant, these are kept separately. Then about 25 to 30 seeds of each of the plant, these of each of the plant, 25 to 30 seeds of each of the plant. These are sown in the separate individual row. Okay, then their performance, it is compared with the existing superior variety, and the rows which are performing better than that of the existing variety, these are kept, while the other rows, these are discarded. Okay, these are discarded. Here also the procedure is repeated same. Then in the last one, that is the best performers, these are then released as the new pure line variety. Okay, here the field technique we have discussed, that is generally it takes about 10 to 11 years to require to produce the new variety. Then the activities which are practiced in the first year we have discussed in detail, that is the large number of the plants are selected from the mixed population. At the time of harvesting, their seeds are picked from each plant, and these are kept separately and numbered. Okay, the seeds are not mixed with each other. Okay, then the next one, in the second year, in the third year, in the second year, that is it gives the second year, the generation of the second year, it will use the experimental strain. Okay, in the third year, preliminary yield trials are carried out. Then fourth to sixth year, main yield trials are conducted. In the seventh year, the multiplication of the improved strain. Okay, at the end of the sixth year, you will get the improved strain. In the seventh year, the multiplication of the improved strain is carried out. And then later on, 8th to 10th year, district yield trials are conducted at different regions, and on the basis of comparative performance, one or two strains are released as the new variety. Okay, we have discussed in detail regarding the procedure which is followed for pure line selection from first year up to the 10th year in detail in the last lecture. Okay, now today we are going to discuss the precautions while practicing the pure line selection in the field. There, the first precaution which we have to take, or the plant breeder have to take, is the mechanical mixtures of the seeds. Mechanical mixtures of the seeds, that is in the field, you know, that is the different types of the crops and the plants are present. Okay, that's why the precaution should be take that the um, seeds of the improved strain should not be get mixed with the seeds of the other varieties of the same or the other crop. Okay, that means the seeds, mechanical mixture of the seeds, that is the seeds of the selected plant should not be get mixed with the seeds of the other variety of the same crop or the other crop. It should be avoided, it should be prevented. ओके, लक्षात आलं. म्हणजे ज्या क्रॉपचं सिलेक्शन केलेलं आहे फील्डवरती, त्या क्रॉपचे सीड्स कुठल्याही प्रकारे दुसऱ्या व्हरायटीच्या, सेम क्रॉपची दुसरी व्हरायटी समजा फील्डवरती असेल, तर त्याचे सीड्स किंवा दुसऱ्या क्रॉपचे सीड्स त्या सिलेक्टेड क्रॉपच्या सीड्स मध्ये मिक्स झाले नाही पाहिजे. That is the first most important precaution, is that the mechanical mixtures of the seeds should be avoided. The seeds of the selected crop must or should not be get mixed with the seeds of the other variety of the same crop or the other crop. It should be prevented. Okay, that is the first precaution. Then the second one, natural cross pollination. Okay, natural cross pollination. Okay, just we have discussed this method, it is generally practiced for the improvement in the self pollinated crop. Okay, but there may be the certain negligible chances of the cross pollination in the self pollinated crops. Okay, and these chances, maybe these are very little chances, but these little chances of the natural cross pollination should be prevented. ओके, लक्षात आलं. म्हणजे प्युअर लाईन सिलेक्शन हे सेल्फ पॉलिनेटेड क्रॉपमध्येच आपण करत असतो. पण काय होतं, कधी-कधी सेल्फ पॉलिनेटेड क्रॉपमध्ये सुद्धा कधी-कधी नॅचरली काही थोड्या प्रमाणात का होईना क्रॉस पॉलिनेशन होतं. जर हे थोड्या प्रमाणात होणारं क्रॉस पॉलिनेशन सुद्धा आपल्याला प्रिव्हेंट करावं लागतं. ओके. नॅचरल क्रॉस पॉलिनेशन in the pure line selection, it is generally practiced for the improvement in the self pollinated crops, but there may be certain little chances of the cross pollination in the uh, naturally cross pollinated may takes place. That's why the very little chances of the natural cross pollination should be avoided. Okay, then the third precaution which plant breeder have to take while practicing the pure line selection is to prevent the mutation. Okay, mutations, these are nothing but the sudden heritable changes which are occurred. These are called as the mutations. ओके. कधी कधी काय होतं, जवळपास इथे 10व्या जनरेशन पर्यंत क्रॉप त्याच्या प्रोजेनीस प्रोड्यूस केले जातात. मग काय होतं, कधी कधी एखाद्या जनरेशन मध्ये सडन हेरिटेबल चेंजेस होतात आणि त्याच्यामुळे काय होतं, तर त्या क्रॉपमध्ये एखादं दुसरंच कॅरेक्टर दिसायला लागतं. तर आपल्याला काय करावं लागेल, असं जर काही सडन हेरिटेबल चेंजेस झाले आणि एक वेगळ्याच प्रकारचे कॅरेक्टर जर त्या प्लांटमध्ये दिसत असतील, तर ते प्लांट तुम्हाला त्या फील्डवरून रिमूव्ह करावे लागतात. Okay, that is the third most important precaution while which plant breeder have to take while practicing the pure line selection is to prevent the mutations. Okay, the certain off type plants may appear in the population, and that's why these off type plants should be removed every year so that spontaneously created mutations may not become the source of variation in the population or the improved strain. ओके, लक्षात आलं. म्हणजे हे जे स्पॉन्टेनियस जे चेंजेस होतात एखाद्या याच्यामध्ये, एखाद्या जनरेशन मध्ये नवीन प्रकारचं कॅरेक्टर इंट्रोड्यूस होऊ शकतं. कशामुळे, तर सडन हेरिटेबल चेंजेसमुळे. तर तुम्हाला प्रत्येक जनरेशन मध्ये प्लांट ब्रिडरला तिथे लक्ष द्यावं लागतं आणि असे ऑफ टाईप प्लांट्स म्हणजे एकदम वेगळेच कॅरेक्टर असणारे प्लांट दरवर्षी रिमूव्ह करावे लागतात, जेणेकरून म्युटेशन हे जेनेटिक व्हेरिएशन साठी त्याला इन्फ्लुएन्स करणार नाही. ओके, ऑफ टाईप प्लांट शुड बी रिमूव्ड in every year so that spontaneously created mutations may not become source of variation in the improved strain. Okay, that is all about the precautions which the plant breeder have to take while practicing the pure line selection. Then the advantages of the pure line selection. The first one is it is the only method to improve the local varieties of the self-pollinated crops. Okay, we have discussed the mass selection method, it is practiced for the improvement in the cross pollinated crop. Okay, while pure line selection, it is the only method to improve the local varieties of the self pollinated crop. Then the another advantage of the pure line selection is that it is easier than hybridization. Okay, this method, the another method which is also used for the improvement in the self pollinated crop, it is hybridization. Okay, but it is pure line selection, it is the method which is easier than the hybridization. Okay, why? In the hybridization there are certain technical operations the plant breeder have to carry out. Okay, that is the first one is the emasculation, and another important factor is the crossing over. Okay, emasculation means what? Emasculation, the removal of a male reproductive organ, stamen or anther, or the killing the pollen grains in the bisexual flower is called as the emasculation. ओके, त्याच्यामध्ये इमॅस्क्युलेशन म्हणजे काय, आपल्याला हव्या असणाऱ्या व्हरायटीचे पोलन ग्रेन्स त्या स्टिग्मावरती टाकून त्याचं पॉलिनेशन करायचं असतं. ओके, त्याच्यामुळे काय, तर त्या सेल्फ पॉलिनेटेड क्रॉपमध्ये त्याचा अँथर किंवा स्टेमेन हा रिमूव्ह करतात किंवा पोलन ग्रेनला मारून टाकतात. त्या प्रोसेसला आपण म्हणतो emasculation. And the prevent the to prevent the random cross pollination, the bagging is carried out. That means in the hybridization, emasculation is carried out, and there is a control on the pollination process. Okay, but in the pure line selection, just we have discussed there is no need to carry out emasculation, and also there is no control on the pollination. Okay, that's why the pure line selection, it is considered as a easier method than that of the hybridization. Okay, that is due to the in the hybridization, emasculation, and there is a control on pollination. These are the two barriers, and that's why it is easier than the hybridization. Okay, then the another advantage is that the variety which is developed, it is extremely uniform in appearance. Why it is extremely uniform in appearance? Because all the pure lines, these are genetically identical. Okay, these are genetically uniform. ओके, त्यांचा सगळ्या प्युअर लाईन व्हरायटीचा जेनेटिक कॉन्स्टिट्यूशन हे सिमिलर असतं. अँड दॅट्स वाय the variety which is developed by the pure line selection, it is extremely uniform in appearance and performance, and therefore these varieties are more attractive. Okay, then this method, it is used in both self and cross pollinated crop for the production of pure lines and inbreds. Okay, understand that this method, it is used for the production of the pure line variety in the self pollinated crop, and for the production of the inbreds in the cross pollinated crop. Okay, these are the advantages of the pure line selection. Then there are, though there are certain advantages of the pure line selection, there are also the certain type of the limitations to this method. Okay, in this one, this method, pure line selection, cannot be used for the development of varieties in the cross pollinated crop. Okay, this method, it cannot be used in the cross pollinated crop because there are certain barriers. This method, it can be used in the cross pollinated crop, but in the cross pollinated crop, as these are heterozygous, the self pollinated, these crops must be subjected for the self pollination for the number of generation. Okay, and that's why the extra attention is required to grow, to practice the pure line selection in the cross pollinated crop. Then it will require more labors to carry out the self pollination in the cross pollinated crop, that is to remove the off type plants or the lower yielding and the dwarf plants. Okay, then the variety which is produced in the cross pollinated crop by using the pure line selection, it cannot be maintained for the number of generation, as it is heterozygous due to the genetic recombination and crossing over. After when this variety, it is cultivated for the number of generation, their characters get deteriorated. Okay, then also the farmers, they don't know anything, they does not have the any knowledge regarding the control on pollination. That is how the cross pollinated crop, these are subjected for the self pollination. Okay, though this method, it can be used for the cross pollinated crops, but it is very difficult to produce the variety in the cross pollinated crops. Okay, that's why we are saying this method, it cannot be easily practiced for the improvement in the cross pollinated crops. Okay, ही मेथड क्रॉस पॉलिनेटेड क्रॉपसाठी सुद्धा आपण यूज करू शकतो. पण काय, तुम्हाला क्रॉस पॉलिनेटेड क्रॉप्समध्ये जर प्युअर लाईन्स तयार करायचे असतील, तर तुम्हाला खूप साऱ्या जनरेशन पर्यंत, कारण क्रॉस पॉलिनेटेड क्रॉप्स काय आहेत, हेट्रोझायगस आहेत. ओके, आणि प्युअर लाईन म्हणजे काय, तर त्या होमोझायगस सेल्फ फर्टिलाइज्ड प्लांटच्या प्रोजेनी आहेत. म्हणजे तुम्हाला काय करावं लागेल, क्रॉस पॉलिनेटेड क्रॉपमध्ये सेल्फ पॉलिनेटेड व्हावं अशा कंडिशन्स तुम्हाला तयार कराव्या लागतील. ओके, म्हणजे त्याच्यामध्ये क्रॉस पॉलिनेशन होणार नाही, फक्त सेल्फ पॉलिनेशनच होईल, नंबर ऑफ जनरेशन. ओके, मग त्याच्यामुळं काय होतं, पुन्हा तुम्हाला एकतर पॉलिनेशनचं कंट्रोल करावं लागेल. पुन्हा प्रत्येक जनरेशनमध्ये, पहिल्या फ्यू जनरेशनमध्ये आपण डिस्कस केलं की ऑफ टाईप प्लांट्स तयार होतात, म्हणजे अल्बिनो प्लांट्स असतील, डॉर्फ प्लांट्स असतील. ओके, तुम्हाला प्रत्येक जनरेशनमध्ये हे प्लांट्स तुम्हाला रिमूव्ह करावे लागतील. म्हणजे त्याच्यासाठी प्रत्येक जनरेशनमध्ये प्लांट्सवरती तुम्हाला जास्त लक्ष द्यावं लागेल. ओके, ठीक आहे. म्हणजे हे थोडंसं जास्त लेबर्स तुम्हाला लागतील मदतीला मग. ओके, म्हणजे थोडंसं एक्सपेन्सिव्ह सुद्धा होईल. ओके. आणि पुन्हा अजून एक आहे, की समजा तुम्ही व्हरायटी तयार पण केली क्रॉस पॉलिनेटेड क्रॉपमध्ये, तर काय होऊन जातं, तर ती व्हरायटी जास्त दिवस ती क्वालिटीची राहत नाही. त्याचे कॅरेक्टर्स हे काही जनरेशन नंतर डेटेरिओरेट होत असतात. ओके, दीज आर द डिफिकल्टीज while practicing the pure line selection method in the cross pollinated crop. That's why there is a limitation. We can say this method, it cannot be used for the development of varieties in the cross pollinated crops commercially. Okay, then this method, it does not develop any new genotype. Okay, that is, we can say there is no possibility of introducing the new character. Okay, as the no new genotype is introduced, we can say the no new character is introduced, which is not originally found in the population. ओके, म्हणजे त्या पॉप्युलेशन मध्ये जर ते कॅरेक्टर नसेल, तर ते कुठल्याही नवीन प्रकारचं कॅरेक्टर त्याच्यामध्ये इंट्रोड्यूस होत नाही. कारण याच्यामध्ये काय, तर कुठलाही नवीन जिनोटाईप याच्यामध्ये तयार होत नाही. फक्त काय, तर जे असलेले जिनोटाईप आहेत चांगले, त्याचं फक्त आपण सॉर्टिंग आउट करत असतो. ओके, म्हणजे कुठलंही नवीन कॅरेक्टर जे की ओरिजिनल पॉप्युलेशन मध्ये प्रेझेंट नाही, ते यामध्ये इंट्रोड्यूस होत नाही. Okay, as there is no possibility of introducing the new character not originally found in population, that is new genotype are not produced. Okay, this method, it is only concerned with the isolation of best genotypes present in the mixed population. Okay, it just deals with the isolation of the best genotypes present in the mixed population. No new genotypes are produced. Okay, then another important thing is that due to the high degree of homozygosity, okay, as the all the pure lines are homozygous, due to the high degree of homozygosity, their adaptability to the varied environmental condition is also pure. Okay, the adaptability or the acclimatization of the variety, it is depending upon the various factors, and one of the important factor is the genetic variation. Okay, more the variety which is having more genetically variable, it get adapted easily to the wider environmental conditions, but the variety which are produced by using the pure line selection are purely homozygous. ओके, ह्या होमोझायगस व्हरायटी आहेत, त्याच्यामध्ये जेनेटिक व्हेरिएशन जास्त नसतं. म्हणजे नॅचरली काय, देअर अडॅप्टिबिलिटी टू द व्हराइड एन्व्हायरमेंटल कंडिशन इज ऑल्सो प्युअर. ओके, देन द another limitation, when we compare this method with the mass selection, we can say it is very lengthy and the laborious process. Why it is lengthy and laborious process? Because in the mass selection we have discussed, that is the plants are selected, these are harvested, and their seeds are collected, and these are mixed or composited together. Okay, these are the seeds of the each of the plant, that is these are not kept separately. But here in this method, the plants which are selected, these are harvested individually. Their seeds of each of the plant, these are kept separately, and these are never mixed up with the seeds of the other plants. Okay, and that is up to the 10th generation. That means up to the 10th generation, the progenies of the selected plants, the plant breeder have to evaluate. ओके, म्हणजे काय होतं, जे प्लांट सिलेक्ट केलेले आहेत, त्या प्लांट सिलेक्शन केल्यानंतर, हार्वेस्टिंग केल्यानंतर तुम्हाला त्याचे सीड्स मिक्स करायचे नाहीयेत. समजा 200 प्लांट सिलेक्ट केलेले आहेत, तर प्रत्येक प्लांटचं हार्वेस्टिंग करून त्याचे सीड्स एका पॅकेटमध्ये तुम्हाला वेगळे ठेवावे लागतात, त्याला नंबरिंग करून झाडाचं नाव देऊन. ओके, हे सीड्स दुसऱ्या प्लांटच्या सीड्स बरोबर मिक्स होणार नाहीत, याची तुम्हाला प्रिकॉशन घ्यावी लागते. हे कधीपर्यंत, जवळपास 10व्या जनरेशन पर्यंत. Okay, that means this process, it requires more time, more space, and that's why it is more expensive. Okay, and that's why we are saying This is the limitation of the pure line selection, that it is very lengthy and the laborious process. Okay, it is very lengthy and laborious process because it requires more time, more space, and thus the process becomes expensive. Okay, these are the limitations of the pure line selection. Okay, then here in brief, you can see the differences between the mass selection and the pure line selection. Okay, here you can see the particulars and then mass particular, or we can say the character in which the mass selection and the pure line selection is differing from each other. Here the nature, we can say the mass selection, it is said to be the more of an art than science. Okay, but in pure line selection it is more of science than the art, because the seeds are not mixed with each other and the progenies of the selected plants are evaluated. That's why the mass selection, it is more of an art than the science, while the nature of the pure line selection is that it is more of science than an art. Then regarding the history, the mass selection we have discussed, it is as old as agriculture itself, but the pure line selection, it is not so old as the mass selection. Okay, then here regarding the number of plants selected in first year, in the mass selection, as far as possible, the large number of the plants are selected in the first year, but in the pure line selection, lesser number of the plants are selected in comparison with the mass selection. Okay, in the mass selection, the large number of the plants are selected in first year, while in the pure line selection, the lesser number of the plants are selected. Then regarding the procedure of the mass selection is that the selected plants, these are mixed together and sown as such in the next year. Okay, just we have discussed in the mass selection, the large number of the plants are selected and the 1 minute please. Okay, एकच मिनिट ना काहीतरी हा yes. Okay, the in the mass selection you can see the selected plants, these are mixed together. Okay, the large number of the plants are selected, these are harvested together, and the seeds of all the plants are collected, these are mixed or composited together in the mass selection. Okay, while in the pure line selection, in this you can see the selected plants are kept separately and the progenies of each plant, these are sown separately in an individual row in the next year. Okay, that means here the seeds are not composited together, and the seeds of each of the plant, these are kept separately and these are sown in separate individual row in the next year. Okay, and here in the number 5, testing of progeny. In the mass selection, the testing of progeny does not carried out. Okay, while in the pure line selection, progeny as well as the individual performance is tested. Okay, progeny test, it is not carried out in the mass selection, while the progeny as well as the individual performance is tested. Then they control our pollination. In the mass pollination, there is no control on pollination. Okay, because the mass selection method, it is generally practiced in the cross pollinated crop, but in the pure line selection there is a control on the pollination. Just we have discussed the precautions which are practiced, which the plant breeder have to take while practicing the pure line selection. Here the very little chances of the cross pollination should be prevented. Okay, then the type of developed variety, as the variety which is developed in the mass selection, it is heterozygous, and as it is heterozygous, due to the segregation of characters, the variety, it get deteriorated very quickly. That is due to the heterozygosity and cross pollination. Okay, cross pollination crop मध्ये जेनेटिक रिकॉम्बिनेशन आणि सॅग्रिगेशन ऑफ कॅरेक्टर्समुळे प्रत्येक जनरेशनमध्ये त्याचे कॅरेक्टर्स हे सॅग्रिगेट होत जातात आणि व्हरायटीचे ओरिजिनल कॅरेक्टर लॉस होतात. Okay, as the variety produced, it is heterozygous and it deteriorates very quickly due to the heterozygosity and the cross pollination. Okay, while in the pure line selection, the variety produced it is homozygous, and therefore it is more lasting. Okay, even if the even if these pure line varieties, these are cultivated for the number of generation, but as these are homozygous, these are more lasting. Okay, they does not get deteriorated very fast. Okay, then the repetition of the process here, the every year, the to maintain the purity, every year this method is practiced. Okay, while in pure line selection there is no need to repeat every year. Okay, regarding the adaptability, as the variety which is produced by the mass selection method, these are heterozygous. Okay, and generally the acclimatization or the adaptability of the new variety, it is depending upon the genetic variability, as the cross pollinated crop or the variety which are produced in the mass selection method, these are heterozygous. These varieties are more suited to the large areas under varying environmental conditions. But the pure line varieties, these are homozygous, and that's why these are not suited or get adapted to the limited region. Okay, these are not having the wider adaptability, or in other word we can say these are adapted to the very limited region. Okay, then for the mass selection, the time taken, that is, it takes about 8 years to produce the new variety by using the mass selection method. And in the pure line selection, it takes about 10 years to produce the new variety. Okay, then another difference we can mention here, that is the mass selection. It is the method which is generally practiced for the improvement of the cross pollinated crop, and while the pure line selection method, it is the method which is used for the improvement of the self pollinated crop. Okay, that is how we can differentiate the mass selection and the pure line selection. Okay, by studying the differences between the mass selection and the pure line selection, you should not get confused between these two methods. Okay, in the examination, the question may be asked for the long answer question may be asked for the 20 mark, as give and detail account on the mass selection and its field technique. Okay, then it may be asked just as give and detail account on the pure line selection and its pure line and its field technique. Okay, then you have to write all these points mentioning in this method. Okay, so you will get the full marks. Okay, that is all about the pure line selection. Okay, just we have completed the two methods of selection. Okay, selection, that is the second method of the crop improvement. Okay, there are four principal methods of the crop improvement. The first one is the domestication, plant introduction and acclimatization. The second one is the selection. The third one is the hybridization, and the last one is the mutation breeding. Okay, of these four methods we have discussed the domestication, plant introduction and the acclimatization, and we are discussing the selection process. There are two types of the selection methods. The first one, natural selection, that is which is naturally operating, and the second one, the artificial selection, that is which is carried out by the human being for their own benefit. There are three methods of the artificial selection. The first one is the mass selection, which is practiced in the cross pollinated crop. The second one, it is pure line selection, which is practiced in the self pollinated crop. Okay, we have discussed in detail these two methods, the mass selection method and the pure line selection method. And the remaining third method, it is the clonal selection method. Clonal selection, it is the method which is used for the improvement of the asexual producing crops, or we can say the vegetatively propagating crops. Okay, that is all regarding the today's lecture, and the third method of clonal selection we will discuss in the lecture which we will take on the Saturday, because tomorrow it is a holiday. Tomorrow is Christmas, that's why it is a national holiday. We will take the lecture on the Saturday. Okay.